I want to give a thank you to our sponsors. So we do have a number of organizations that support us throughout the year, and I do have a quick slideshow that I'll throw on in the breaks. You can kind of have a view of who's supporting us. We do welcome new supporters. So if you know anybody that's interested in either sponsoring or supporting the organization as a business, we would encourage that. And also we do have options for subscriptions. So if you're interested in the information that we have to offer, you can get a digital subscription. You'll get a free, like a no pass to our 2 digital events. If you're interested in the in-person events, we have an agronomy subscription that allows you to come to all of our in-person events. And we're super excited for a new 2-day field school that sort of takes us back to our roots. And it should be lots of fun in the field and lots of hands-on learning opportunities, which we're super excited about. And if there's anybody that has suggestions on topics or things that they really want to learn about this year, feel free to send me a note on that as well. We're still finalizing the agendas, and there's a little bit of space still. So if you have any ideas that would be really great hands-on sessions, I'm all ears and would love to find a speaker to match any of your needs. If there's any questions, like I said, don't hesitate to reach out. Is there anything that I didn't cover that anybody needs an answer to before we get rolling this morning? Thanks, got it. Okay. I believe Charles is on the line, on the call.
I'm here.
Perfect. Awesome. Welcome, Charles. Okay, we'll get maybe started a few minutes early and give ourselves a little bit of a buffer. So Charles is a research scientist here at the AAFC in Lethbridge, and he's focused on herbicide-resistant weeds in Western Canada. And this morning he's going to present on herbicide-resistant diagnostics, where do they fit and how can they help? And we'll allow Charles to turn on his presentation, and I'll let you take it from here.
Okay. All right, I should be sharing my screen there. Can you just confirm that?
Yes.
Okay, excellent.
Hello.
All right, okay. So yeah, thanks for the introduction. My name is Charles Geddes. I'm a research scientist in weed ecology and cropping systems with Agriculture and Agri-Food Canada, based out of Lethbridge, Alberta. And today I'm going to be talking about herbicide resistance diagnostics. We're going to jump right into the weeds here. So I'll be covering 4 main points. The first is I'll give a bit of a background on herbicide-resistant weeds in Canada. I'll talk about some of the diagnostic labs that are available to us across the Canadian prairies. And then I'll talk about 2 main groups of herbicide resistance diagnostics called whole plant bioassays, which is the most common, and then also genetic testing, which is sort of a new type of resistance diagnostics that we're working on. I should mention that our research program, it's called Weed Ecology and Cropping Systems, but broadly we focus on herbicide resistance, where we look at discovery of new types of resistance across the Canadian prairies, characterizing new biotypes, looking at where they occur on the prairies, and then trying to develop strategies to manage those issues as well. So to jump right into it here, I'm starting off with definitions. So this is the definitions of herbicide resistance versus herbicide tolerance, mainly because the 2 seem to be used interchangeably sometimes, and that's actually not the case if you look at their definitions, where herbicide resistance is the inherited ability of a plant to survive and reproduce following exposure to a dose of herbicide normally lethal to the wild type. And so the key there is that it's something that has evolved in a plant community, and it is inherited among generations. When you compare that to herbicide tolerance, it's the inherent ability of a species to survive and reproduce after herbicide treatment. So this implies that there was no selection or genetic manipulation to make the plant tolerant. It is just naturally tolerant. So to give a couple examples here, so for herbicide resistance, I'll talk about kochia because that's my favorite weed. So for example, glyphosate resistance in kochia. So glyphosate resistance, we know what causes resistance. It's a mechanism called EPSPS gene duplication, and we'll get into that later on in the presentation. But it is something that evolved in kochia, and it is inherited among generations. And we selected for this type of resistance with recurrent use of glyphosate. When you compare that to herbicide tolerance, though, an example could be, for example, the use of group 1 herbicides in our small grain cereal crops. So we'll use wheat as an example. Wheat is naturally tolerant to group 1 herbicides, even though these herbicides are used to manage grass weeds, and wheat is a grass crop. But wheat naturally metabolizes these group 1 herbicides. And so it is something that has always been present in that species. We didn't necessarily select for herbicide resistance. It just so happens that that species metabolizes those herbicides rapidly. So to take a global perspective here, this is showing the number of unique herbicide-resistant weeds by country. And in Canada, we could say we're in 3rd place when it comes to herbicide resistance, as far as having the greatest number of unique herbicide-resistant weeds. So behind the United States, which has the most, and Australia, which has the 2nd most, Canada is in 3rd place. And so certainly part of that is due to land base under agricultural production and a predominant focus on herbicides in our cropping systems. But yeah, in Canada, we're in 3rd place. And so when we look at the discovery of new types of herbicide resistance over time, this is showing the first discovery of unique herbicide resistant weeds. And so when I'm talking about a unique herbicide resistant weed, what I'm referring to is the combination of a weed species resistant to a herbicide mode of action. So for example, group 1 resistant wild oat would be a different unique biotype than group 2 resistant wild oat. And so we can see resistance actually started in Canada back in the 50s. And on the prairies, it was more so in the 80s when we started seeing herbicide resistance showing up and increasing at a steady pace, essentially, as years have progressed. It's certainly not slowing down any, but we're sitting at 86 unique herbicide-resistant weeds across the country, with a rate of discovery of 2 new types of herbicide-resistant weeds each year. And so increasing herbicide resistance is of course important when we consider that we have a limited number of herbicide modes of action to manage these weed issues. This figure is showing the release of new herbicide sites of action or modes of action over time. And you can see at first glance, it looks like, wow, there are a lot of different herbicide modes of action that we have to manage weeds. Some of the main ones that we use on the prairies are highlighted there in red. But what I point out with this figure is that we essentially see a plateau starting in the 80s, where we've gone on about 4 decades now and only had 1 new herbicide mode of action released. And unfortunately, that mode of action has no uses in prairie agriculture, at least to date anyway. So essentially, release of new herbicides these new herbicides tend to be reformulations of older herbicide modes of action or different combinations of different active ingredients, but they're not new modes of action. So we'll jump into question number 1 here. So can we pull up question number 1? Can everyone see question number 1? Yes.
It should show up both on screen with a pop-up and also in the chat if you don't happen to see the pop-up.
Okay. I can see it on my other screen, but it's not on the right screen. And I don't have the option to move it, so you may have to look in the chat for it. So the question is, which 2 herbicide groups have the greatest selection pressure for herbicide resistance? And in this, you'll have different options of different herbicide groups or modes of action. and you should be selecting 2 of them. The options are group 1, 2, 3, 4, 5, 9, 14, and 15. Jamie, can you confirm if you can see the poll?
I can see the poll. I do see we have 51 responses so far. And do you want to know the percentages?
I can see them now because I
Okay.
filled it in myself. But so we'll move on here. So what I can see, at least so far from the 58 responses, is that we have 24% for group 1, 41% for group 2, and then we're sitting at about 30% for glyphosate or group 9. And so the correct answer here is actually group 1 and group 2. And so we'll move on here. This is just a figure that was put together by Hugh Becky back in the early 2000s, showing the selection pressure for herbicide resistance. And so some of the group numbers have changed a bit, but group 1 and group 2 herbicides tend to have the greatest selection pressure for herbicide resistance, especially when it comes to target site resistance. And so this is a figure that I put together that outlines the different herbicide resistant weed species that are present on the Canadian prairies. And so you can see there's different types of herbicide resistance that are known to occur in the region. With group 1 here, we have 5 different group 1 resistant grass weeds present on the prairies. We have about 23 different group 2 resistant, both grass and broadleaf weeds. Many people are surprised to see the large number of group 2 resistant weeds that we have present on the Canadian prairies. We also have resistance to group 3 in green foxtail, 4 different species resistant to auxin mimics or group 4, 3 glyphosate resistant weeds, for example. And so there are many different types of herbicide resistance that farmers are dealing with on the Canadian prairies. And so this is just a list of what I consider to be some of the top issues that farmers are dealing with on the Canadian prairies. And it's not in any specific order, but some of the bigger issues that we have in our production systems are group 1 and 2 resistant grass weeds that are causing a reliance on soil applied herbicides. So an example there would be group 1 and 2 resistant wild oat, starting to be quite common on the Canadian prairies and causing reliance on soil applied residual herbicides like triolate or peroxysulfone, for example. The next is spread of multiple herbicide-resistant kochia. Kochia is known to exhibit resistance to up to 5 different herbicide modes of action, and you can see these traits stacking together, causing multiple resistance to different modes of action. Advancing range of invasive amaranthus species, so namely water hemp and Palmer amaranth. So both of those species have been found in Manitoba. And water hemp, especially in Manitoba, is spreading a bit more quickly. But we do expect these weeds to be moving further into Western Canada from Manitoba, probably starting off in eastern Saskatchewan. And so both of those species are devastating a lot of crop production systems in the United States. And they can exhibit resistance to almost all of the products that have been registered to control them. Next, we have the use or reliance on glyphosate at multiple use windows, predominantly due to the fact that we have glyphosate-resistant crops, but then we also have widespread adoption of reduced or no-till systems. And finally, spread of several group 2 resistant weeds, those 23 different species that I talked about earlier. So let's pull up another poll here. And we'll go with question number 2. So the question that you should be able to see now is, what percentage of annual cropped fields in Western Canada have herbicide-resistant weeds present? So your options are 10 to 20%, 30 to 40%, 50 to 60%, 70 to 80%, or 90 to 100%. So fill out the poll and we'll see what you think. Okay, I'm going to fill out the poll as well. Hopefully you can't see my answer, just so I can see the results here. And so the majority of people, sitting at 41% right now, are saying 50 to 60% of annual crop fields have herbicide-resistant weeds present. The next closest one was 30 to 40%. And so the answer here is actually 70 to 80% of the fields, at least based on our surveys of herbicide-resistant weeds across the prairies. And so this is one of the projects that I have the pleasure of leading. It's called the Prairie Herbicide Resistant Weed Surveys, and we survey over 800 randomly selected fields across the prairie provinces over a 4 to 5 year time frame. And in this particular project, the way it works is we tend to do a different province each year, except for Saskatchewan is over 2 years because of the larger land base there. And what we do is the fields are randomly selected quarter sections, and the number of fields within each eco district is stratified. So based on the agricultural land area within that eco district. And so once we identify the quarter sections, we then contact the farmers, ask for permission to access the fields that were selected. And then for the resistance part, we visit those fields right before crop harvest. And what we're doing is we survey the field and look for visible uncontrolled weed patches soon before crop harvest. And so we visit those weed patches, collect, if they have mature seed, we collect mature seed from those patches. We also note the location of the patches and estimate their area as well. And so we bring those samples back to our lab where they are tested for different types of herbicide resistance, mostly group 1 and group 2 resistance, using whole plant bioassays. And so what we can do is we can look for different types of herbicide resistance in a whole range of weed species. And that's where we can aggregate the information together to come up with some of these metrics, like the percentage of fields that are occupied by herbicide resistant weeds. And you can see how that has been changing over time with the different within the different provinces. And so this is just an example of the Saskatchewan survey that took place last in 2019 and 2020, and we'll be starting again with Saskatchewan next growing season, I guess this year in 2024. So in Saskatchewan, we surveyed 419 fields, and what we found was that 74% of the fields where we collected and tested weeds had some form of herbicide resistance present in those weeds, and that equated to about 72% of all fields under annual crop production in the province. So that's where we come up with that estimated percentage of fields. And so we pair this survey also with a grower management questionnaire. It asks questions about management practices that have taken place within those fields, but then also other questions like what is the perceived cost of herbicide resistance and reduced crop yields and quality and increased weed control expenses. And so we can aggregate this information together to come up with the cost of herbicide resistant weeds for Saskatchewan farmers, which is currently estimated at about $340 million a year. And so in the Saskatchewan survey, some of the most common species that we're picking up, so this could be viewed as weed species that are left uncontrolled before crop harvest within those fields. And so some of the most common broadleaf weeds are kochia in Saskatchewan was the most common one that was picked up, followed by wild buckwheat, lamb's quarters, Canada thistle, and sow thistle. Mainly this is spiny sow thistle or spiny annual sow thistle. Then for grass weeds, not surprising that wild oat was the most common species picked up, followed by green foxtail, barnyard grass, foxtail barley, and annual brome species like downy brome or Japanese brome. And so some of the results here, these figures are presenting the results on a percentage of fields. So this would be the percentage of all 419 fields that were surveyed that had this type of herbicide-resistant weed present. So for example, with group 1 resistance here, 46% of fields in Saskatchewan had group 1 resistant wild oat present, followed by 8% for green foxtail and 1% for yellow foxtail, which is just starting to move into the province from Manitoba. For group 2 resistance, you see we have a whole range of different species resistant to group 2. Most commonly was kochia in 39% of the fields in the province, followed by wild oat or group 2 resistant wild oat in 18% of those fields, and then a whole range of different weed species between 1% and 4% of the fields in the province. When we look at multiple resistance in wild oat to group 1 and 2, we're sitting at 16% of those fields in Saskatchewan. So for the Manitoba survey, we covered 155 fields in the province, and a lower number of fields than Saskatchewan due to the lower area under agricultural or under crop production. But what we found was that 75% of all fields in Manitoba had herbicide-resistant weeds present. And so this equated to an annual cost to farmers in the province of $81 million a year. So some of the species that were left uncontrolled before crop harvest in Manitoba, as a percentage of the fields where they were collected here. So for broadleaf weeds, pigweed species tended to be show up the most in the survey. This was mainly redroot pigweed, but there was also some green pigweed, or what we call Powell amaranth, as well as smooth pigweed showing up on the survey as well. Followed by lamb's quarters, wild buckwheat, kochia in number 4, and then pale smartweed. For grass weeds, again, wild oat showed up the most, followed by green foxtail, barnyard grass, yellow foxtail, and foxtail barley. So when we look at the resistance as a percentage of all fields under annual crop production in the province, again, for group 1 resistant, wild oat was the most abundant, followed by green foxtail, yellow foxtail, and so on. For group 2 resistance, wild oat again was the most abundant, followed very closely by group 2 resistant barnyard grass in Manitoba, and then kochia in 19% of those fields, and pigweeds in 13%, and so on, a whole range of other species at 10% or less of the fields in the province. And group 1 and 2 resistant wild oat showed up in about 30% of the fields under annual crop production in the province. And so moving on, that's just a quick overview of some of the most recent round of the resistant weed surveys. And so I'll talk a little bit about diagnostic labs for resistance testing now. And so this information was taken from an infographic that was put together by the Resistant Wild Oat Action Committee. And so some of the prices may have changed because this was a couple of years ago now. But what this infographic does is it highlights the different types of tests that are offered by different labs across the prairies. So to start off here with the Crop Protection Lab out of Regina, Saskatchewan, what they offer is whole plant bioassays for a whole range of biotypes that are known to occur across broad acreage on the prairies, like group 1 and 2 resistance in a whole range of species, followed by some of the soil applied herbicides as well. Group 8, which is now group 15, would be triolate, or group 3, which would be trifluralin there. If we look at AgQuest out of Minto, Manitoba, they offer similar tests that are whole plant bioassays for a whole range of different types of resistance that are known to occur on the prairies. The Pest Surveillance Initiative out of Winnipeg, Manitoba, they offer a test for glyphosate resistance in kochia, but their test is a molecular or a genetic test. So we'll get into the difference between the whole plant bioassays offered by the other labs and this genetic test that's offered by the Pest Surveillance Initiative. And so the fourth lab here is our research lab out of Agriculture and Agri-Food Canada in Lethbridge. And unlike these other diagnostic labs that do sort of a fee-for-service structure, our focus is more on the research end and documenting new types of herbicide resistance on the prairies. And so we offer free testing, but only for weeds that are suspected to have novel or new types of herbicide resistance in the region. And I'll get into a few examples of these, but the idea is that this offers, it kind of works both ways, where we confirm for, say, a farmer or an agronomist that yes, they may have this new type of herbicide resistance that is showing up. It allows us to start some research on figuring out how to manage those issues as well. And it gives us more information on the status of herbicide resistance across the prairies. So question number 3 now. So the question is, have you used a diagnostic lab for herbicide resistance? It should be herbicide resistance testing instead of training. But have you used one of these labs in the past? Yes, no, or maybe? I'll just give you a minute to fill it out there. Okay, so we're sitting at about a 40-60 split at the moment, where we have 40% ish are saying yes, they have used one of these labs in the past, and 60-ish% saying no, they have not. So we'll move on here, and we're going to get into a little bit more about how we sample for these diagnostic tests and the different types of tests that are offered here. So this is just kind of a general recommendations for sampling for submitting samples to diagnostic labs. And so it comes with this asterisk here at the bottom that it's important to follow, to visit the website for whichever lab you're planning on using, and follow their sampling and submitting protocols. But in general, the way that whole plant bioassays work anyway is that the labs need to have mature seed from the weeds that survived a herbicide application in the field. So general recommendations would be to collect about 2,000-ish mature seeds from about 10 to 20 different plants that survived the herbicide application in the field. So this essentially, you have to let those weeds grow, go to seed, and then collect the seed from there. And so then air drying those seeds and packing them in a paper bag or an envelope, preferably not plastic, to help avoid any spoilage of those weeds while they're being shipped. And then mail or ship that sample to the lab of your choice. So we'll talk about whole plant bioassays. So I've mentioned this a few times now in the presentation. And so most of these groups, what they do is they conduct a single dose whole plant bioassay. And this is just an example for glyphosate resistance in downy brome here. And so what we see on the left is our susceptible control sample. So this would be susceptible downy brome. On the right is our resistant control sample. And then in the center is a sample that was submitted by a farmer or an agronomist. And the idea is that they take those mature seeds, plant them under controlled environment, grow up those plants, treat them with a herbicide, and then they look at their phenotypic response to the herbicide as the herbicide is having its effect on the weeds. And by comparing it to resistance and susceptible controls, you can see certainly this submitted sample looks like it is resistant to glyphosate. And so some of the results that we get back are what I call resistance incidence, or the percentage of plants that have the resistant trait. So for example, say 100 plants grew in this sample, and 90% of them survived the herbicide. So we would have a resistance incidence of 90% in that sample. So some labs would give you that 90%. Other labs would just give you a general grouping, basically binning the percentage into susceptible, which would be 0% versus low, moderate, and high resistance based on that resistance incidence percentage. And so the reason why some labs just give this general grouping is because these percentages can quite easily be misinterpreted, where a farmer can think, oh, I maybe only have 15% resistance in this particular sample, which means that I can get away with applying that herbicide and still see 85% control. And it's not quite that simple, right? And that's not really the recommendation that we're going with for these percentages that you're getting back, because a population can very quickly go from 15% to 85% resistance in a single growing season, right? So it would make more sense to strategically save that herbicide for application down the road when you really need it, right? So that's kind of why those percentages can be misinterpreted. So the next, so I talked about single dose bioassays, which are offered by a lot of these diagnostic labs. And so when we have a new type of herbicide resistance showing up and characterizing that type of resistance, we need to have a bit more information to have an official first confirmation, because the rates that you're choosing for your single dose bioassay can vary, and they're not always based on the field rate of that herbicide. And so this is more of an example of what we do in our lab for the first confirmation of a new type of resistance, where we do a dose response experiment and subject the populations to a whole range of very low to very high rates of the herbicide. So this is just a visual example of one replicate of a dose response experiment. This was for PPO inhibitor or group 14 resistant kochia from Saskatchewan. And we have our resistant sample in the back there, and then two susceptible controls in the middle and the front, and exposed to a whole wide range of rates of saflufenicil. A rough field rate would be this 25 here. And so you can see we're going up to 100 times that field rate. And so when you analyze these data, you get this, you have to fit these nonlinear regression models. And so the way that you design the experiment actually plays a large role into how well those models actually fit in the end. So it takes a lot of background knowledge to actually design the experiment appropriately and design the right rate structure. But this is sort of what you get in the end. We have biomass fresh weight as a percentage of the untreated control here. So 100% would be similar to the untreated control. And you look at how the response of that population changes over a wide range of rates. And so what we do is we pull out where this dashed line crosses there. This is known as a GR50, or the rate of the herbicide required to reduce biomass by 50%. And so we pull that out for the resistant population and the susceptible populations. And then we compare the resistant to the susceptibles and just calculate a ratio of those. So what we observed with this particular population was that it exhibited 70 to 92.5-fold resistance to foliar applied saflufenicil. And so this is the globally accepted method to confirm a new type of herbicide resistance. If we're uploading a new case to a global database, for example, we have to provide this information to the manager of that database in order to have it uploaded as a new case that's present in the region. And so the beauty of these whole plant bioassays is that they work regardless of the mechanism that confers herbicide resistance. And so something that we talk about maybe a bit less is that there's multiple different mechanisms that can confer herbicide resistance in weed species, right? And so that's kind of what we're showing here, organized into two broad classes. There's target site resistance and non-target site resistance. And so I'll provide a few examples of each of these. So I mentioned whole plant bioassays work regardless of the mechanism that confers resistance, and that's why they are considered one of the standards for resistance diagnostics. And so genetic testing focuses specifically on target site resistance, which just so happens to be the most common type of herbicide resistance that we deal with across the prairies, but also globally as well. So the difference between the two, or the main difference, is the timing that you can get results back from each type of testing. So this is just showing 2 calendar years here. We'll say that a post-emergence herbicide is applied in June. The farmer or agronomist is scouting, say, 2 to 3 weeks later to see how well that herbicide controlled the weed that they were targeting, and they noted lack of control at that point. So if you went with genetic testing, it's as simple as collecting leaf tissue from those uncontrolled weeds and submitting it to a lab that can get you a result back in 1 to 2 weeks. Whereas if you go with the traditional whole plant bioassays, that means you have to let those weeds grow, go to seed, collect the mature seed, submit that to a lab, and then the testing happens throughout the winter, essentially. And so it's really the timeline of the two that's different, where genetic testing allows you to make additional decisions in the same growing season, while whole plant bioassays require you to wait and make a corrective action in the next growing season. So here's just a few more differences between the two types of resistance testing. So for genetic testing, it requires leaf tissue from the plants, whereas whole plant bioassays require mature seeds. For genetic testing, we have to know the mechanism that confers that type of resistance in the first place, whereas whole plant bioassays work regardless of the mechanism of resistance. Genetic testing is based on target site resistance, whereas whole plant bioassays work for any of the resistance mechanisms. One requires a molecular lab versus a greenhouse or a growth chamber and a controlled environment sprayer. Whereas the other main thing is the level of diagnostics that you're getting. So genetic testing will give you plant level diagnostics. So is that resistance mechanism present in those plants that you sampled? Whereas the whole plant bioassays give more of a population level, where I talked about that resistance incidence, or the percentage of individuals that survived the herbicide when it was applied. And again, the biggest thing is the timeline of the two tests, where one is more of a rapid test, the other one takes quite a bit longer. So we have a project that was funded by ARDAR to develop these genetic tests for herbicide resistance. And so over the past few years, we've developed or adapted 14 different genetic tests for different herbicide-resistant weed biotypes that are present on the Canadian prairies. And so the tests target specific types of herbicide resistance. So we have to understand what are the mechanisms at the genetic level that confer resistance in the first place. And so for these genetic tests, the idea is that we're also working on developing a kit for sample submission for genetic testing for herbicide resistance. And I'll get into this in a bit more detail here. So the idea is that you're scouting your field after herbicide treatment, and you see an uncontrolled weed, you suspect that it may be herbicide resistant. So you sample that particular species. We're talking about nice lush green tissue, about 3 to 4 inches in size, and that tissue would then, obviously you're recording the species that you're sampling and the crop that it was in, and so on, and certainly the suspected type of resistance or the herbicide that was applied in that field. Those samples would then just go in a simple Ziploc bag with these silica gel beads that help to desiccate the weed sample. And so it's a fairly simple diagnostic kit that we can put together for genetic testing here. And the idea is that each bag could have a different plant sample in it, going into a larger Ziploc bag that would represent a field. and again, outlining some of the identification information where the results should be sent. And you can have different kits for each field. And so it's a pretty simple procedure. And the one of the cool parts of it is that these beads actually change color when they take on moisture. And so you can tell if the kit is still viable, first of all, and if those beads are not viable, it's as simple as just putting them in the microwave and drying them down, and they should return to orange again, indicating that they're viable. And so it's recommended to submit a sample in 1 to 2 weeks, but obviously the faster you submit your sample, the more rapid you'll get a result back. So when we're talking about processing samples now, what happens is when the sample arrives at the lab is that we're extracting DNA from the leaf tissue. Here just showing the DNA extraction. Here we're looking at the quality of the DNA that was extracted from the tissue. And we are developing PCR-based tests to identify specific mechanisms at the genetic level. And so you can come up with tests like this, for example, that are separating our susceptible samples from our resistant samples. And so the result that you get back is whether that specific mechanism or the resistance conferring mechanism was present in those samples that were submitted or not. And so we know, based on other research, that certain mechanisms are associated with different patterns of cross resistance. So by knowing the mechanism that confers resistance, we can have an idea of which herbicides are not effective, but also which herbicides should remain effective in that species as well. So I'll go into a little bit more depth here, but I'll keep it at a fairly broad level. We'll talk about a target site mutation here. And so at a very simplified level, the way a herbicide works is you have your active ingredient here shown in green, and your active ingredient, when it's taken up by a plant, it binds to an enzyme target site, and that active ingredient essentially shuts down a certain step in a pathway that's required for growth and development of a plant. And so the particular pathway that's targeted varies depending on the herbicide mode of action. So we have our herbicide binding to the target site and effectively helping to shut down that particular pathway. However, if you have a target site mutation now, you have a genetic mutation in the sequence that codes for this enzyme target site. And this mutation can alter the structure of the target site, for example. And now your herbicide is not as easily able to bind to that target site. So it either doesn't bind or it has reduced binding affinity. And so an example here, we'll talk about a bit of a case study. Over the last couple of years, we've been working on group 1 resistance in foxtail barley. So the particular cases that we were working with were submitted from the Peace River region in Alberta. And so we started off with this dose response experiment. So we had 3 what we called putative resistant populations from different fields submitted to the lab, comparing them with 2 susceptible controls here to a whole range of quizalofop. So the farmer and agronomist in this case suspected that these foxtail barley populations were quizalofop resistant. And so showing some of the data here, on the far left, we have a dose response with quizalofop here. And what we found was that 2 of those 3 putative resistant samples exhibited between 15 and 29-fold resistance to quizalofop, whereas the 3rd putative resistant sample was actually susceptible because it had a ratio that was close to 1, meaning that it's responding similarly to the herbicide as our susceptibles. If we look at clethodim now, so clethodim is another group 1 herbicide, but from a different chemical family within group 1, we found that all of these ratios were similar to 1. So these particular populations were resistant to quizalofop, but susceptible to clethodim. We also note this interesting, what we call a hormesis peak here. I'm not going to get into that, but it really doesn't play any role in this resistance confirmation. So just an artifact of low dose of herbicides that are applied. So we went on to do some Sanger sequencing of the ACCase gene within these plants. Here we have 3 susceptible plants on the top, 3 resistant plants on the bottom, and this is just showing a diagram of a partial sequence of ACCase. The idea is that the different colored peaks here are calling for different nucleic acids, and if we compare our susceptible plants to our resistant plants, if you look really closely, we have double peaks here for our resistant plants, suggesting that there was a change in a nucleic acid there. And so we can develop a genetic test to identify that, specific mutation. And it just so happened in this case that these plants were heterozygous for this target site mutation, which is known to confer group 1 resistance in other grassweed species. So for the second type of target site resistance is increased expression of target site genes. And I'll get into an example of this specifically on gene duplication. So we go back to our active ingredient here, applied. It's supposed to bind to this target site, but with gene duplication now, we have a duplication of the region that encodes for that enzyme target site. So now we have multiple target sites that over recurrent generations are increasing within that population. So now when you apply the field rate of the herbicide, it can bind to the target site, but it can't bind to all of them. So the pathway continues either unobstructed or less obstructed in this case. So for case study 2 now, we'll talk about glyphosate resistance in downy brome. So I mentioned this a bit earlier. The story here is that back in 2021, an agronomist suspected that they were dealing with glyphosate resistance in downy brome, noting that the downy brome was uncontrolled in glyphosate-tolerant canola. And so the sample is submitted to our lab. We conducted the dose response experiment. Here is an example shown at the bottom, where we have our resistant population in the back compared with 2 susceptible controls in the middle and the front, exposed to a wide range of rates of glyphosate in this case. And some of the data that came out from that in the top corner here, this is survival 3 weeks after treatment with glyphosate. And what we found was that our resistant population, shown here in blue, exhibited between 10 and 12-fold resistance to glyphosate. So it certainly was surviving those field rates of glyphosate that we're applying. Digging into it further, we did similar research looking at sequencing. EPSPS is the target site for glyphosate. And so we did sequencing. We didn't note any resistance conferring mutations. But when we conducted qPCR to look at the number of copies of the target site, we actually found that there was a large difference here. So in the bottom right here, we're showing an EPSPS to ALS ratio. So ALS here interestingly is a target site for group 2 herbicides, but it's used as a housekeeping gene here because it's relatively stable. And so we're looking at the relative copy number of EPSPS to ALS in these plants, noting that about 29-fold increase in EPSPS copy number in our resistant plants compared with our susceptible plants. And so this is really tightly correlated with the LD50 for glyphosate, or the rate of glyphosate required for 50% plant mortality. So we know that EPSPS gene duplication is associated with glyphosate resistance in downy brome. And so this also happens to be a similar mechanism for glyphosate resistance in kochia. Here on the right, I have the EPSPS to ALS ratio. So as we get further away from 1 here, it means we have an elevated number of copies of EPSPS, and that is associated with the LD50 for glyphosate in kochia as well. And so this is the only known mechanism for glyphosate resistance in kochia to date. So I'll talk about a little bit about non-target site resistance now. I'm not going to go in depth into each of these examples, but for reduced absorption, so this is a mechanism that is reducing the amount of the herbicide that's actually absorbed by the plant. I won't give an example for that though. Reduced translocation or vacuolar sequestration. So this is the herbicide is being taken up by the plant, but it's not moving as easily within the plant. So what can happen is that the plant actually sequesters the herbicide active ingredient in a vacuole in the plant cell, and that herbicide is not able to actually reach the target site in the end. The third one here is increased or enhanced metabolism, and I'll talk about this one a little bit. But the whole point is that these non-target site mechanisms are mechanisms that are reducing the ability for the herbicide active ingredient to reach the target site in the first place, whereas target site resistance is a modification of that target site. So again, we have our active ingredient and our herbicide binding site. But before that herbicide even reaches the target site, it's being degraded by the plant's metabolism. And that the metabolism of the plant breaks that active ingredient down into less phytotoxic metabolites that are not able to have the same effect on that enzyme target site. So for metabolic resistance here, this is just comparing a target site mutation versus metabolic resistance. And so we'll talk about a target site resistance or mutation here first. So these mutations are naturally occurring within species. They're not caused by the herbicide, but they're selected for by herbicide application. So you can have a very random and infrequent mutation that just so happens to confer resistance to a particular herbicide. Then a farmer applies that herbicide, and because that plant has resistance, that is one of the only species or one of the only plants that are growing and producing seed. That seed goes into the seed bank, and now you have a population with that mutation next year. And that's sort of how it builds in the population. These mutations generally confer a higher level of resistance initially, but in a lower number of plants, and the number of plants with the mutation then builds within the population. It's selected for by recommended doses of the herbicide or even high rates of a herbicide. And it has a lower potential for cross resistance between modes of action because your mechanism is related to a certain enzyme target site. So it's related to that specific mode of action. And in this case, herbicide rotation can help to help mitigate this type of resistance or manage it if it does show up. For metabolism-based resistance, however, it's more of a slower build of resistance in populations where it often it starts off slowly, and you're selecting for plants that have a higher and higher rate of metabolism. And so the resistance tends to be lower level at the start, and it's building at a population level. And it's selected generally by lower rates or non-lethal rates of herbicides. So if you're applying herbicides at reduced rates, this is one of the biggest risks, is selecting for metabolism-based resistance. And so the idea is that if you're using a lower rate of the herbicide, you're obviously not seeing the same level of control, and the plants that are surviving that herbicide are the ones that have a higher metabolism to degrade the herbicide active ingredient. So as this happens over time, you can see how the metabolism would be increased within that weed population. It has a higher potential for cross resistance, and so the biggest issue with metabolism-based resistance is that you can select for resistance by applying one herbicide, but it actually confers resistance to a whole range of different herbicides, and it can be cross modes of action in this case. And it can even be selecting for resistance to modes of action that aren't even available on the market yet. So that is the big scary thing about metabolism-based resistance. And in this case, rotating herbicides will be less effective because it's not as specific to a mode of action. So everyone asks about wild oat. And so my answer is always it's complicated because Wild oat is known to exhibit both target site-based and non-target site-based resistance. So this is just some work done by Hugh Becky, and the dark, if the red color is darker here, it means that incidence of resistance is higher, or there's a greater percentage of individuals in that specific population that had the resistance trait. And so on the far right here are the various mechanisms that were identified to confer resistance in those populations. We have our ACCase inhibitors. APP would be your FOPs, this would be your DIMs, and this would be your DENs. So different chemical families within group 1, and then you have your a couple different families within group 2 as well. And the whole point here is just to demonstrate that there's multiple different target site mutations that confer both group 1 and group 2 herbicide resistance in wild oat, as well as some populations with enhanced metabolism that confer more of a broad spectrum of resistance within those populations. And you can even have stacking of target site-based resistance and enhanced metabolism in wild oat. So this is why it's complicated. This is some research also by Hugh Becky back in the early 2000s. And what we're looking at here this is different products within group 1. So this would be your FOPs, 3 products there, and then your DIMs here. And again, the percentages are the resistance incidence. So 100% means all of the individuals had that resistance trait. 0% would mean it's susceptible. And so within wild oat, you can see these different patterns of cross resistance that are conferred by all of those different target site mutations that can confer group 1 resistance in wild oat. And so I note again, these data are over 20 years old now, but you see all of these zeros for clethodim. Things, first of all, don't misinterpret that. We certainly do have clethodim resistance on the prairies, but it tends to be at a lower frequency than some of the other group 1s. Similar for pinoxidin, which isn't shown here, we tend to see a lower frequency of resistance to pinoxidin. But the point with this is that there's variable cross resistance, and this is why resistance testing is important, because it can identify which herbicides are no longer effective in your field, but also which ones are effective in your field. And so if we look at the crop protection guide here, there at the start of the crop protection guide, they do have tables outlining different herbicide groups and the different products that are located within different chemical families within modes of action. So this can be used to help select products that to which your wild oat population is still susceptible, and this is similar for other weed species as well. So this is a really important resource to use when selecting your products to use in your field. And so just to end it off here, we know that resistance is a growing issue on the prairies, and largely it's due to an oversimplification of our weed management programs. Over the past decades, we've essentially been spoiled where herbicides have been highly effective to manage weeds, and I mean, it's of course farmers are going to focus on a highly effective and efficient form of weed control. Even when we have resistance present, the first things that farmers go to manage those resistant weed issues are mixing, rotating, and layering herbicide modes of action. And cultural and physical weed management take more of a supplementary role in contemporary production systems. We do need to start moving more toward an integrated weed management approach that integrates physical, cultural, biological, and chemical weed management together in order to get ahead of this issue. And certainly it doesn't have to be this complicated, but this is just outlining that there are various options for alternative weed management that should play more of a forefront role in our weed management systems moving forward, at least in order to help mitigate herbicide resistance. So with that said, we know that testing for herbicide resistance can be done through different methods. Most of the labs currently on the prairies offer whole plant bioassays, but we're building the capacity for genetic testing on the prairies. And so in the end, it's really whichever method and whichever type of resistance testing works best for your situation. Do you want a result rapidly, or do you want more of a broader picture of resistance regardless of the mechanism that's conferring it? And so it's really what works best for your situation. So with that said, I'd like to thank all of the funders who generously support our research on herbicide resistance, as well as the collaborators, and it takes an entire research team to do this type of research, and it's really all of their work that I'm presenting on today. So thanks very much for listening, and I'd be happy to answer questions.
Okay. Well, thanks so much, Charles, for your great presentation. As always, I always learn something. It's inevitable when you give a presentation, so thank you. If you have questions, feel free to type them in the chat. Alternatively, you can raise your hand. There were a few questions that came into the chat early, so I will just share those now. So first question was, is there an even distribution to resistant weeds across the provinces, or do they follow any specific patterns of production or environmental factors?
Yeah, so that's a really good question. And so the answer is it depends, I guess. So some weeds we know that they tend to be abundant across the prairies. For example, wild oat, right, is pretty much everywhere where we have annual crop production on the prairies. And we tend to see group 1 and group 2 resistance, for example, across that entire range of area. Other species, however, tend to be more isolated, right? You can see how it would resistance when it is first evolving, starts off isolated, and then spreads from there. Part of it is related to how quickly that resistance spreads. For example, in kochia, it can spread very quickly because it's spread through both seed and through pollen. But other species are selfing, and it doesn't spread quite as quickly that way. So it really depends on the weed and the biology, as well as the specific mechanism of resistance. We do see certain types of resistance that are more isolated to certain geographical regions on the prairies, but then others, it's very widespread. So it's really tough to do sort of a broad generalization across species and types of resistance in that case.
Yep, that definitely makes sense. Next question, is there herbicide resistance data for Alberta, and maybe is it available publicly online?
Yeah, so I always get this question. So we sampled Alberta in 2023 in the fall, and so we have to wait for those seeds, first of all, to after ripen to help alleviate dormancy. Then the resistance testing starts in the winter. So we're really only starting that at the beginning of that resistance testing for those Alberta samples, but we will have those results become available here over the next year. So all of the processing and diagnostics that happen after we collect the samples usually takes about a year for that province before we have the data available.
All right. Another question is anyone looking at ways to overcome or reverse any of the mechanisms for resistance, increased rates, or other means?
Yeah. So I guess overcoming resistance, it can be difficult. So first of all, what herbicide resistance tends to be conferred through it's basically a dominant gene, right? So or most types of resistance. So it's not as simple as using, say, a refuge to reintegrate susceptibility back into weed population. If you, say, put out susceptible kochia in a field, will that help revert your resistant plants back to susceptible? And the answer is generally, unfortunately, no. What will happen is you're just turning those susceptibles you put out into resistant ones because it's dominantly inherited, right? So it's not quite as easy as some other pests. When it comes to overcoming resistance through management, of course, that happens by applying, say, alternative modes of action or using, say, cultural weed management strategies. But in general, when we start to see resistance showing up in a field, it's very difficult to completely get rid of it, unfortunately. So that's why mitigating these issues makes a lot more sense, although it requires an economic investment before you have the issue present in your field, and that's sort of the big crux and the issue of herbicide-resistant weed mitigation and management.
All right. And is there any Group 9 resistance foxtail barley in southern Alberta?
Uh, the answer is no, not that we know of. So our lab requires, we rely on farmers and agronomists to submit samples of lack of control to our lab so that we can look into these issues for you. And so we haven't had that show up in any samples that have been submitted so far. It's not to say that it doesn't occur, but we haven't found it anyway.
All right. And there's one final question that is absolutely unrelated, but we did have a request for you to play a song, perhaps Smoke on the Water by Deep Purple.
Oh, wow. I'll have to start charging if we get into that.
And thank you, Charles, very much for your presentation. I think
Thanks.
there's lots of folks here that are also saying thank you in the chat.
Yeah, thank you for the invite and for your attention here over the last hour. Much appreciated.
Yeah, thank you so much.